This method can help answer key questions in the C. elegans field, such as cell biology. The main advantage of this technique is that it is fast and efficient to obtain signals of nuclear staining, GFP, and subcellular localization changes simultaneously.
To begin, generate an extrachromosomal array line with a translational construct of target genes. Alternatively, obtain such lines from the Caenorhabditis Genetics Center, which is a public resource for C. elegans research, or from a previously published research laboratory. After exposing worms to gamma radiation to integrate the extrachromosomal lines into the genome, select the stably expressed lines so that each animal expresses a marker protein, such as a GFP or roller. To remove mutations generated during the radiation, outcross the lines at least 2 times. Use the resulting transgenic line to detect protein expression pattern changes under stress. Next, after preparing NGM worm plates according to the TEXT protocol, streak an OP50 clone and culture the bacteria in liquid LB broth overnight. Seed the NGM plates with liquid OP50 culture and incubate the plates in a 37°C incubator overnight to grow a bacteria lawn as a food source for the worms. Cool down the plates at room temperature for at least 30 minutes before use. Grow the transgenic line at 20°C without starvation.